All right, welcome to the Explainer. Today we are getting into the absolute core of what makes life, well, life. We're talking about the fundamental building blocks of everything, amino acids. Let's just start with this idea from Francis Crick, one of the guys who helped crack the code of DNA. What he's saying here is just incredible, that these 20 little molecules are basically the same everywhere, in every living thing. It's a simple thought, but it's got huge implications. Just let that sink in for a second. 20. That's it. This tiny alphabet is responsible for all the amazing diversity of proteins you see on this planet. The stuff that makes a jellyfish glow, the silk in a spider's web, the very enzymes that are working in your body right now, it's all written with just these 20 letters. It's wild. Okay, so here's our game plan. We're going to start with the big picture, then zoom right into what these things actually look like. We'll take a tour of what makes them different, talk about where we get them, see how they get put together, and then find out what happens after they're assembled. All right, let's get right into it. First up, what exactly are amino acids, and why are they so ridiculously important for, everything that's alive? So at its most basic, an amino acid is a building block. Honestly, the best way to think about them is like biological Legos. You take these little bricks, you snap them together in different orders, and you can build basically anything. From the complex machinery that digests your lunch to the keratin that makes up your hair. These long chains of Legos have a technical name, polypeptides, which is really just the raw material for what will become a fully formed protein. Okay, so we've got these biological Legos, but what do they actually look like up close? If they're the universal building block, they've got to have some kind of shared design, right? And yep, you guessed it, they absolutely do. Every single one of those main 20 amino acids has the same core structure. Right in the middle, you have the alpha carbon. Attached to that, you have three standard parts. An amine group, that's the amino part of the name, a carboxyl group, that's the acid part, and a hydrogen atom. But that fourth spot, that's the R group. Now that's where the magic happens. It's the one part that's different for each one, and it gives each amino acid its unique personality. So because that central carbon is holding on to four different things, you get this really cool property called chirality. All it means is that they exist as two mirror images of each other. It's like your hands, they're mirror images, but you can't stack them perfectly on top of each other. We call them the L form and the D form. And what is absolutely fascinating, for reasons we are still trying to figure out, is that life is a total lefty. It almost exclusively uses the L form to build proteins. The D form is super rare, though you can find it in some weird places, like the cell walls of bacteria. So we've got our 20 standard letters that our DNA spells with. But biology just loves to break its own rules, doesn't it? Turns out there's a plot twist. There are actually two more that are much rarer, selenocysteine and pyrrolysine. And they're so special they don't have their own code. The cell has to perform this sneaky little trick where it basically sees a stop sign in the genetic code and says, nah, I'm going to run this red light and pop in one of these special guys right here. Now, let's get back to that R group, because honestly, this is where all the action is. This is the part that gives each amino acid its unique flavor, its chemical character. It's the source of all the incredible variety we see in the world of proteins. Now, 20 different R groups might sound like a lot to keep track of, but we can actually make it easier by sorting them into families based on their behavior. You've got your nonpolar ones, which are hydrophobic, they hate water. You've got some with a negative charge and some with a positive charge. Then there are the aromatics with these big ring structures, and others with hydroxyl groups, which are perfect for certain chemical jobs. And this whole water-fearing versus water-loving thing, it is a huge deal in biology. Our cells are basically bags of water. So when you make a long protein chain, what happens? All the hydrophobic, water-fearing R groups panic and try to hide from the water by tucking themselves into the middle. And all the hydrophilic, water-loving ones, they're perfectly happy to hang out on the surface. This simple push and pull is one of the biggest forces that makes a protein fold into its final, beautiful, functional shape. And don't think for a second that these are just tiny chemical details. The real-world consequences are absolutely massive. Take hemoglobin, the protein that carries oxygen in your blood. Swap just one water-loving glutamic acid for a water-fearing valine, and you get sickle cell disease. It's all because that one change makes the proteins clump together. The disorder PKU is caused by the body's inability to handle phenylalanine. And the special features on lysine and serine make them perfect targets for modifications that act like on-off switches, controlling what the protein does. Okay, so these molecules are obviously vital. We need them. So, where do they come from? That brings us to a really practical way of looking at amino acids.
This is where we get into essential versus non-essential amino acids, and that naming can be a little confusing, so let's clear it up. Non-essential are the ones our body can make itself. It's got the factory for it. Essential are the ones we have to get from our food. Our bodies just can't build them. Now, the key thing to remember is that essential doesn't mean more important. They're all critical. It just means it's essential that you eat it.
And another thing, this list isn't set in stone. It's actually pretty flexible. What's essential for a person might not be for, say, a dog. It can change as we get older. And sometimes, if someone is sick or under a lot of stress, their body might not be able to make enough of a non-essential amino acid. In that case, it becomes conditionally essential. You temporarily need to get it from your diet. Okay, so we've got our supply of amino acids, either from our food or made fresh in our cells. Now for the really cool part. How do we actually snap these Legos together to build something? The special connection that links amino acids is called a peptide bond. And this thing is like biological superglue. It's a really strong covalent bond, which means it's incredibly stable and holds things together tightly. It's formed when the carboxyl end of one amino acid joins up with the amine end of another. And when that happens, they have to spit out a water molecule. The whole process is so elegant. You just line them up, the carboxyl end of one meets the amine end of its neighbor, a little molecule of water leaves the party, and bam, you've got a peptide bond. That gives you a dipeptide, two linked together. And then this just happens again and again and again, adding one more link to the chain each time, building this long string based on instructions from our DNA. We have different names for these chains depending on how long they are. A short one is an oligopeptide. A really long one, we're talking hundreds or thousands, is a polypeptide. And what's really, really important is that this chain is a one-way street. It has a distinct beginning, which has a free amine group, we call that the N-terminus, and it has a distinct end with the free carboxylic group, the C-terminus. That direction matters a lot. So you might be thinking, okay, the chain is built, job done. But no, not even close. For so many proteins, this is just where the story really gets interesting. These amino acids have a whole life after they've been put on the assembly line. Think of the basic protein chain like a brand new smartphone. It works, sure, but the real power comes when you install apps. That's what these post-translational modifications are. They're like little chemical apps that get added to the amino acids after the chain is built. You can add a phosphate group to act as an on-off switch. You can add a hydroxyl group to make collagen super strong. You can attach sugars or other little tags to completely change the protein's job. It massively expands what those 20 letters can do. And as if all that wasn't enough, amino acids have some pretty important side hustles too. They're not just for building proteins. That feeling of well-being from the neurotransmitter serotonin? Yep, that starts with the amino acid tryptophan. That rush you get from dopamine? Thank you, tyrosine. Even histamine, the molecule behind allergic reactions, starts as histidine. They're not just building materials, they're also fuel, and the starting point for some of the most critical molecules in our bodies. So, this all leads to a really mind-blowing question, and it's where science is headed right now. If nature managed to build this entire, incredible, complex world with just 20 letters in its alphabet, what could we build if we started adding new ones? Scientists are already doing it, creating new medicines and materials. It makes you wonder. Life wrote the first book with 20 letters, but the story is far from over. What are we going to write in the next chapter?